All right, still in chapter 24, picking up with slide 109, and we're at the next accessory organ, which is the pancreas. Now, the pancreas is a leaf or feather-shaped organ. It's underneath the stomach, and it extends from the duodenum, which is the first section of the small intestine, towards the spleen, which is behind the stomach. Now, it's also retroperitoneal, which means that it is outside of the peritoneal cavity. It won't have the peritoneal membrane on the outside of it. So instead, it's wrapped in a thin connective tissue capsule. There are going to be quite a few organs that are going to have a capsule, including the liver and the kidneys and the pancreas. And we already saw a capsule on the adrenal gland. So it has a head, a body, and a tail. The head is closest to the intestine. It's the widest part. The body, it gets a little narrower and goes out. And then the very tip of it, which is short and rounded, is the tail. And running through the center of it is a rather large, prominent tube called the pancreatic duct. Now, up near the head, the pancreatic duct splits, and we'll see that in just a few seconds. So what is the pancreas made of? Remember from the endocrine chapter that it has both endocrine and exocrine function. We've already talked about the endocrine function of the islet cells, like the beta cells, the alpha cells. The rest of the pancreas, which is really the bulk of it, is divided up into lobules, and then each lobule is made up of clusters of these acini, pancreatic acini or acinar cells. They are little blind pockets made up of simple cuboidal epithelium. That's the exocrine part. So they're going to create digestive enzymes. The pancreatic islets are the patches of endocrine tissue that are scattered all along the acini, and they are only about 1% of the pancreas cells. So here's the liver, here's the first section of the small intestine known as the duodenum, and here is that feather or leaf-shaped pancreas. This round part that sits in the curve of the duodenum is the head. And then this is the body, and then here is the tail. And you can see running through the center the pancreatic duct. And as we get close to the head where it's going to interact with the small intestine, it splits into two separate ducts. We have the regular pancreatic duct here, and then we have an accessory pancreatic duct. So we have two ways of sending the secretions made by the acini cells into the small intestine, into the duodenum. And this is showing you again the pancreatic duct. This would be an islet. So we have all of our endocrine cells and then surrounded by these acini made of simple cuboidal cells. And here it is in a photomicrograph. Again, the endocrine parts will secrete insulin and glucagon. The exocrine parts are going to make several different enzymes that are going to create an alkaline pancreatic juice. Now, why does it need to be alkaline? So it can counter the acidic secretions as the chyme leaves the stomach, okay? So about a liter of a day is secreted by the pancreas, and it has digestive enzymes, water, and ions, and it's controlled by hormones that are coming from the intestine, the first section, the duodenum. So the enzymes that come out of the pancreas are pancreatic alpha amylase. So you can tell if it's amylase, that's going to help digest carbohydrates. Pancreatic lipase, which is going to digest lipids. Nucleases. What in the world would a nuclease be needed for? Well, in your food, there's DNA and RNA. So we have these nucleotides we need to digest down to. So that will take care of those. And proteolytic enzymes is going to attack proteins. So we have our carbs, our lipids, our nucleic acids, and our proteins. So there are enzymes for all four macromolecules coming out of the pancreas. Pancreatic alpha amylase is a carbohydrase, an enzyme that breaks down carbs. It's almost exactly the same as salivary amylase. Pancreatic lipase is going to start breaking down lipids or fats. And it's going to release fatty acids, which remember are the building blocks of lipids. And they're going to be easily absorbed. Nucleases break down the RNA or DNA in the food. Proteolytic enzymes break apart proteins. So we have proteases for big proteins and peptidases for smaller amino acid chains. About 70% of all the enzymes produced by the pancreas are going to be proteolytic enzymes. And like we saw in the stomach, they're secreted first as an inactive proenzyme, and then they'll have another thing that will cleave them and activate them into the active enzyme. The proenzymes that are coming from the pancreas And remember, if it ends in ogen, you take the ogen off when you cleave it. So trypsinogen is going to be converted to trypsin. Chymotrypsinogen is going to be chymotrypsin. Procarboxypeptidase, we take off the pro, it becomes carboxypeptidase. And proelastase, it just becomes elastase. Okay? And notice, we get the active trypsin, and then trypsin activates these other ones. So once we've activated trypsinogen to trypsin in the duodenum, the trypsin can activate these other protein enzymes. All right, so that was it for the pancreas. The liver is the largest of your visceral organs. It's in the right hypochondriac and epigastric regions, and it can even go into the left hypochondriac and umbilical regions. It has over 700 jobs, and it performs essential metabolic and synthetic functions. So, looking at the liver itself, it has a tough fibrous capsule. It's also covered with visceral peritoneum. It's divided into a right lobe and a left lobe that are really obvious. And then we have two smaller lobes that you have to look at it from the inferior side to actually see them well. They are the caudate and the quadrate. And the right and left lobes, which are easy to see, is because there's a falciform ligament that shows you the division between them. And at the back, it gets thicker, and then it becomes what we call the round ligament. Now, we have a bunch of blood vessels that are going to converge at what we call the Porta Hepatis. Hepatic will tell you you're in liver territory, okay? What happens is all of your digestive system is going to absorb the nutrients from your food. And the very first place all of those nutrients are going to go is to your liver. Now, a portal system is, again, a circulatory system that is for doing something other than delivering oxygen and picking up CO2. It's kind of like a separate circulatory system within an organ. And for the liver, we're going to bring in all these nutrient-rich blood supply into the hepatic portal vein. So all the nutrients are going to come into the liver, and then the liver is going to deal with and direct the nutrients to where they need to go. So about a third of the blood supply is arterial blood coming from the hepatic artery proper, and the 2/3 is venous blood coming from the hepatic portal vein. Now inside, the cells that do the work in the liver are called hepatocytes, and they are going to be controlling the levels of nutrients circulating through the body by absorbing some and secreting others. Okay, so here is a nice superior abdomen view. Okay, here is the falciform ligament right here, or excuse me, here. Here is the Porta Hepatis, where we're going to have all the blood vessels and stuff going in. And this is a really hard view to look at. Let's look at this view. This is better. Okay, so we have our left lobe. This is the right side of the person's body. So left lobe, right lobe. Okay, and the falciform ligament gets wider and comes out as the round ligament. And then this little thing peeking out from under the right lobe is the gallbladder. Okay, so this is a separate organ, but it is associated with the underside of the liver. If we look at the underside, the back of the liver, okay, so here's our gallbladder, here's the inferior vena cava, here's our left lobe and our right lobe. The other two lobes are here and here. Okay, so this one is kind of formed where the liver rubs up against the inferior vena cava. And you see how this kind of looks like a little animal tail? The word caudate means tail-like. So this is the caudate lobe, this little part here. And then here is this part, looks like a little diamond shape, like a quadrilateral. And it's formed between the round ligament and the gallbladder and the porta hepatis and the inferior edge. This little quadrangle is the quadrate lobe. Okay, the gallbladder is here, and we're going to talk about this more, but we have the hepatic portal vein entering here. We have the hepatic artery that enters here, and we're going to have the ducts that lead to the gallbladder and beyond. Okay, so we'll get to the ducts later. So inside the liver, we're going to have tons of connective tissue that's going to divide it up into 100,000 lobules. And so a lobule is like the smallest working piece of the liver. So we're going to take the hepatocytes and we're going to arrange them in a hexagonal shape to make a lobule. And we're going to have a central vein, And then we're going to have a bunch of sinusoids. Now remember what sinusoids are. They're the leakiest of the types of capillaries. So we have the hepatocytes forming plates, and they're arranged like the spokes of a wheel. The sinusoids are between them, and they all empty into a central vein. And we're going to have lots of macrophages here because, again, anytime we have stuff coming in through the digestive system, we might allow pathogens in. So these are going to line the sinusoids and make sure that nothing bad is entering the circulation of the liver. So here's what they look like. Okay, so this is one liver lobule. Okay, and it's kind of six-sided hexagon. Right in the center is the central vein. Okay, and then you have the plates of hepatocytes, and you have the sinusoids. This would be these very leaky capillaries that are going to the central vein. So that's what's going in the middle of the lobule. Now, at the corners, for all six corners, we have what's called a portal triad. And the portal triad has an artery, a vein, and a bile duct. Get this to move. Here we go. Central vein, okay? Here are our sinusoids, our capillaries. We have some interlobular bile duct, interlobular vein, and interlobular artery form our triad. So we're going to be bringing a lot of the blood in, right? We're going to be sending it through the veins. We're going to be collecting some of the nutrients and storing some and secreting others. And then we're going to put it out into the circulation. We also see the little tubes where the bile is forming and draining from the liver are called the Bile Canaliculi. That's a word we've seen a couple times already. So again, the portal triad has the interlobular bile duct, the interlobular vein, this time that has blood in it, and an interlobular artery. So the hepatic portal system is dealing with all of the nutrients that have just been absorbed by the digestive system. So each of the six corners has a portal triad. It has six sides, and those are the components of the triad. The liver is absorbing nutrients and secreting bile. Now, fats have a really hard time being broken down into small enough pieces for the enzymes to work on them. So we have to emulsify fats. And so we break them down into teeny little spheres called micelles by adding bile. So it helps break down fats. We make it in the liver, and then we're going to store it in the gallbladder. And we'll secrete it from both the liver and the gallbladder when we detect a high amount of fat in the food that's entering the small intestine. So the liver is actually creating the bile, and it puts it into the bile canaliculi, which will eventually drain into the bile lobules. And the right and left hepatic ducts take all the bile from all the bile ducts, and they're going to come together and make the common hepatic duct. Remember, hepatic means liver. Then the common hepatic duct, it's going to take the bile, and it's either going to go into the regular bile duct, which is going to go into the duodenum, or it's going to go into the cystic duct, which leads to the gallbladder. So the bile duct is made by the common hepatic duct joining the cystic duct. And it goes through the wall of the duodenum, and we inject the bile into the intestine, into the pancreatic duct at the duodenal ampulla. We need a picture. Okay, so here are the two lobes, the right and left lobe. So this is my left hepatic duct. This is my right hepatic duct. Once they fuse in this part of this Y shape, this is my common hepatic duct, and it stays the common hepatic duct until here. Now this is the cystic duct. The cystic duct leads to the gallbladder. So bile is made and it gets collected in the gallbladder, and the gallbladder holds on to it. Okay? And later on, the gallbladder can squeeze bile. Bile can come from the liver directly or through the cystic duct. And once the common hepatic duct and the cystic duct fuse, we just call this the bile duct. The bile duct goes behind the pancreas, and it kind of cheats and joins the pancreatic duct right at this little three-dimensional little bean shape, little donut. That is the pancreatic ampulla, the duodenal ampulla, where it joins the pancreatic duct and the bile duct, and we're going to secrete the pancreatic enzymes and the bile into this section of the duodenum. So here's where they come together. Bile duct, pancreatic duct. We have the duodenal ampulla, which is this enlarged area, and the area that sticks up is the duodenal papilla. And there's a sphincter in there, the hepatopancreatic sphincter. And so we control the bile coming in and the pancreatic juices coming in. And there's the gallbladder. You can see how it joins. Here's the left hepatic duct, right hepatic duct, common hepatic duct. Here's the gallbladder. So the cystic duct is kind of hard to see. It's right here. And then this would just be the bile duct, and that would go down to the duodenum. So we make the bile in the liver, store it in here. So the liver is constantly secreting bile, about 1 liter a day. We take some of it and store it in the gallbladder, and the longer it sits there, the more concentrated it gets. Okay, so if you don't take it out of the gallbladder, it might even crystallize, and then you'll get what? Gallstones. Now, it's waiting for the food in the duodenum to arrive, and if there's a lot of fat in the food, then the enteroendocrine cells of the duodenum will secrete cholecystokinin. Cholecystokinin will go up to the gallbladder and the liver and tell them to release bile into the bile duct so that that bile can help break up the lipid droplets, the fat drops. So people often say, well, people get gallbladder stones if they eat a high-fat diet. Well, actually, that doesn't make sense, because if you're eating a high-fat diet, you're actually going to empty the gallbladder more often. It's actually people who go on a low-fat diet and the bile builds up inside the gallbladder and doesn't get used that actually suffer from more gallstones. So, liver has over 200 functions in metabolic regulation, hematological regulation, and bile production. All blood leaving the digestive system absorptive surfaces has to go into the hepatic portal system, which takes it into the liver. So this is a vein. The hepatic portal vein brings the blood only from the digestive system into the bottom of the liver through the Porta Hepatis. Now, the hepatic portal vein is made by fusing the inferior mesenteric vein and the superior mesenteric vein. They fuse to bring the blood into the hepatic portal vein. Then the liver cells will take all the nutrients and toxins, that's important, from the blood, and then it will take and remove and store excess nutrients. So it will correct nutrient deficiencies by taking the ones it sees that it needs to send out and taking extras and storing them, or making things that it senses that the body needs. So the liver is going to have a lot of different types of receptors for different chemicals and ions and nutrients. So it also controls carbohydrate metabolism, lipid metabolism, amino acid metabolism, waste removal, vitamin storage, mineral storage, and inactivation of drugs. Very, very busy organ. Also, since it had all this metabolic function, it's going to need a lot of oxygen. It gets about 25% of the total cardiac output. It's one of the organs that gets the most blood. It also controls some hematological regulation, meaning blood. It controls phagocytosis and antigen presentation. That's an immune function. It synthesizes your plasma proteins. It removes circulating hormones. It removes antibodies, and it removes or stores toxins. All right, and then again, of course, again, the bile. So, production and functions of bile. Bile salts are used to break up large fat droplets in a process called emulsification, which makes them small enough to be absorbed into lacteals. Increases the surface area exposed to enzyme. It's necessary because all the mechanical digestion in the stomach only leaves large drops of lipids, and enzymes can only work on the surface of the droplets. So we need to make the droplets as small as possible so that the enzymes can get to them and break off the individual fatty acids. Enterohepatic circulation is taking the bile salt between the liver and the small intestine. Okay, the gallbladder. We already seen it. We had an introduction. It's a hollow pear-shaped muscular sac whose only job is to store and concentrate bile. It doesn't make the bile. It just holds it until it receives the signal of cholecystokinin to secrete the bile into the bile duct. And it also has 3 regions, a fundus, a body, and a neck. The fundus is the rounded portion, the widest part. Then comes the narrower body, and then the very narrow neck before it joins the cystic duct. The gallbladder will release bile into the duodenum through the duodenal ampulla only when stimulated by cholecystokinin. Without it, there is a hepatopancreatic sphincter and it will stay closed. So opening the sphincter allows the bile to enter. Bile exiting the liver and the common hepatic duct enters the cystic duct, and since this sphincter is closed and it can't enter the duodenum, it backs up the cystic duct to fill the gallbladder. When the chyme leaves the stomach and enters the duodenum, if it senses a high amount of fat, cholecystokinin is released, which causes that sphincter to open. The gallbladder contracts and squirts the bile into the duodenum to dissolve the fat in the food. A full gallbladder can hold about 40 to 70 milliliters of bile, and the longer it sits, the less water is in it, and so the bile salts and other parts become more and more concentrated. All right, so that's a good place to start, and I will pick up the next presentation with slide 146.